the Sustainability Management Master's Program, you're answering one simple question. Do you care about your future generation? If you care about your kids, their grandkids, then you care about sustainability. And a sustainability manager is someone who has the tools to make people understand how they can contribute towards developing a sustainable world. We have both part-time and full-time students. Our curriculum is 30 hours. Instead of thesis, we have a research project course and a seminar course which provides them with the kind of research background they will need to be successful in their job. But at the same time, sustainability is not always everything about environment. It also relates to the business. They will take a sustainable business strategies course. They will take a project management course. Most of these courses are going to be taught by industry people, the people who are doing sustainability on a daily basis as a part of their job. Any organization that has a large number of employees and has a physical infrastructure, they will have to have a sustainability office. If you have the passion to develop and maintain a sustainable world, come to us and we will help you shape your passion into a career which will create an impact.
University. It's in the Upper Peninsula of Michigan on Lake Superior, actually closer to Canada than to the United States, I guess. So currently she is on a sabbatical, a year-long sabbatical, working at Stevens in the Center for Environmental Systems. Her expertise is developing environmental remediation methods using plants and microbes and their interactions. She was instrumental in developing the bio and phytoremediation programs at Michigan Tech and at the University of Texas at San Antonio, where she was an assistant professor before she joined Tech as part of their strategic faculty hiring initiative in sustainability. I think it was back in 2009. So Rupali has authored or co-authored like more than 250 scientific publications, including journal articles, book chapters, and conference proceedings. She co-edited 2 books with me, Concepts and Applications in Environmental Geochemistry, which was published by Elsevier in 2007, and then An Integrated Approach to Environmental Management, which was published by Wiley in 2015. Rupali got her BS, MS, and PhD in Life Sciences from the University of Hyderabad in India. She did her postdoctoral work at the University of Niigata in Japan, the University of Florida, and then the University of Texas. So today, Rupali is going to talk about sustainable remediation technologies for lead in urban soils. Welcome, Rupali.
Thank you, Dibs. So let me see if I can, okay. So today I'm going to talk about a couple of technologies, the main one being phytoremediation. And so both these technologies, they are sustainable, environment-friendly technologies, and how we went through the different steps of developing this technology is the sort of the topic of my talk today. So lead is a problem that both Dibs and I have been working on for the past 20 years. And there are various sources of lead that in the environment, and the main one in this country is the lead-based paint in the older homes. So I'll talk a little bit more about that. But other than that, also lead pipes is a major problem, and also leaded gasoline. In addition to that, antique pottery, Christmas ornaments, ceramic ware, lead crystal glassware, all of these antique things contain lead. And also some of the imported toys, jewelry, cosmetics could have lead. And then from time to time, we hear about, apple juice or applesauce having lead. So it's pretty widespread contamination and pretty well known. So in urban soils, which we are dealing with, the 2 main culprits are, number one is the lead-containing paint in older homes. So lead-based paint was banned in the US in 1978, but before that, all the lead paint had all the paint had lead in it. And so you can see that the peeling and chipping of this paint would contaminate the yard. And from there on, this lead could be just dragged into the house, converted into dust, and then people could be exposed to it. And although this was banned in 1978, there are lots of homes that are older homes in many cities which still have lead-based paint. And then the other one is the leaded gasoline, which was banned in 1996. And then since then, we know that people's blood lead levels have been going down. So it has made a big difference in lead exposure in people. So here's a slide showing the pathway of lead in the environment, and you can see mainly that industry and mining, of course, and then some pesticides, fertilizers, particularly in New Jersey, the apple orchards where they use lead arsenate as one of the pesticides, and then, of course, the lead-based paint, and then leaded gasoline being the main culprits, leaded pipes. And so from there, the lead gets into soil, water, sediment, and air and food, and people get exposed to it. So the health effect of lead is mainly particularly problematic in children under the 6. They are the especially vulnerable sort of group to this lead because they are still growing, their organs are still growing. So even in low levels, it causes reduced IQ, learning disabilities, attention deficit disorders, growth problems, behavioral problems, damage to various organs. And then, of course, at high levels, it causes more severe problems. So when I talk about lead, I'll have to talk a little bit about the Flint water crisis, being from Michigan. So most of you might have heard about the Flint water crisis. So this was in 2014. The city of Flint actually switched their water supply from Lake Huron to Flint River, and from then on, their water started becoming just worse and worse, and they could actually see the weird smell and the color. And if you Google Flint water crisis, you'll see this picture. Everybody will come across it, and you can see visibly how the water was deteriorating. And at the same time, doctors reported that children's blood lead levels were going up measurably, and so this became a full-blown crisis. And on the right, you can see here that a professor in Virginia Tech, he did this experiment where he just put a nail in the Flint water for 1 month, and you can see what it's looking like within a month. So at a much smaller level, we also had this problem in Newark around 2017, 2018. But here the city government was much more proactive, and they were able to, and also the local nonprofits, and they were able to sort of prevent a full-blown crisis here. But lead toxicity, CDC, their reference elevated blood lead level in children was 10 micrograms per deciliter for a long time. But then in 2012, after a lot of lobbying, a lot of scientists showing a lot of data, they finally lowered it to 5 micrograms per deciliter. However, I want to sort of emphasize that there is no known safe level of lead in the blood. So even 5 micrograms is toxic, and even it's been shown that 3 micrograms is toxic. So lead is not safe at any level at all. So our main concern is the soil lead, although the lead in the drinking water grabbed the headlines, but soil lead is a much more widespread problem all over the country because there are so many homes that were built before 1978, and these homes are the main source of elevated blood lead level in children in this country. And as well as in urban areas, lots of people, they don't have large yards, and so they do use community gardens, and so to grow their vegetables, and that could be also a source of lead. And what is the traditional method people use of remediating these houses is that basically dig and haul, dig up the surface soil and haul it and get rid of the soil in hazardous landfill. And so for residential purposes, this is really unrealistic, very expensive. And so our goal was to develop a more sustainable and a more inexpensive remediation method. So dig and haul being expensive, we could think of two methods. One is phytoremediation, the other is chemical. The chemical remediation work is new, it's going on in our lab. Currently, phytoremediation is the one that I'm going to describe in more detail, and the plant we use is vetiver grass. So phytoremediation, basically you can think of plants as sort of a solar-driven pump, as you can see in this picture on the right, is that the plants, they are able to, along with the water, they are able to pull up the contaminant into their tissue, and then you can gradually harvest the plant and slowly lower the contaminant levels in the soil. So that's the sort of theory behind this. And you don't need any power source for this, plants acting like solar-driven pumps. So that is the beauty of this method. So I'm going to go through the method development of how we developed this chemically catalyzed phytoremediation method through laboratory, greenhouse, simulated field studies, and then we implemented the study in people's homes and in a community garden, which I'm going to show you. So this slide shows why phytoremediation is a useful method. So if you're thinking of remediation of a particular area, so let's say 10 acres. So if you are removing the surface soil and you have to dispose of all of that soil, you can think of the mass of it would be about 30,000 tons. If you are able to grow plants and the plants have taken up that contaminant, and you're only disposing of the biomass, then the amount of the mass that you have to deal with, it comes down right away to 1,200 tons. So that is 25 times less. And then if you're able to ash that biomass, then it further reduces by another tenfold. So this is significant reduction in mass, and that translates to significant reduction in cost as well. So it is basically the reason why phytoremediation is not only environment friendly, but also cost effective. So when we started thinking about developing a method, the first question is, what plant are we going to use? And so looking at the literature at that time, I had found sunflower, and they were using corn, and a few other couple of plants for removing lead. These were mostly food plants or oilseed plants. And so I wanted to see if there is a grass that people could grow easily in their yards. And that was the main sort of focus of our research, is how easily could it be translated to urban homes. And so for that reason, I came across this grass called vetiver grass, the scientific name being Chrysopogon zizanoides. This plant, I found that it's widely grown in mining areas in South Africa, Australia, and also Asian countries. And to prevent erosion, that's the reason they were using it, because the plant has a huge root system. They can hold on to the soil. And also, World Bank was promoting the use of this particular plant. So we didn't know at that time that it was a hyperaccumulator of lead. We found this later. But At that time, we knew that this plant would be fast growing. It has a huge root system. The main consideration being native of Asia, that this plant is non-invasive. So that was a major consideration for us. It's non-invasive because the variety that we use, called the sunshine variety, it does not set seeds. So doesn't produce seeds, doesn't produce stolon, and doesn't sort of grow horizontally either. So USDA labeled it as non-invasive, which was good for us. It's a perennial grass. And again, like I said, it was used for erosion reduction in a variety of countries. So we thought we would check this plant out. And then as we did our laboratory studies, we realized that it not only tolerates lead, but it also is a hyperaccumulator, meaning that it is able to accumulate more than 1,000 milligrams of lead per kilogram of tissue. That's sort of the definition of a hyperaccumulator. So the chemically catalyzed part, why is that important? So if we are using phytoextraction, that is using a plant to extract the metal from the soil, then the metal has to be soluble. So the plant is not going to be able to take up metal if it is tightly bound to the soil. So the first important thing is for the metal to be solubilized or mobilized. So the mobilization part, particularly for lead, is difficult because lead is tightly bound to various soil components. And so the mobilization needs a special kind of treatment. So here I'm showing a 6-month-old vetiver root, and you can see already that the roots are extensive. And then this is a famous picture. You can see how mature roots are so extensive and just so long and huge. So it has a huge biomass, grows very fast. So phase 1 was our laboratory incubation study, where we wanted to check out what kind of chemical could we use to mobilize the lead. That is a chelating agent. So when we talk about chelating agents, EDTA is a very well-known, widely used chelating agent, and it's in the literature. It's known widely to mobilize lead, but then it's highly effective. So we did not want the lead to be mobilized too fast, where either it becomes toxic to the plant, or it gets into surface water and the pollution is spread further. So we didn't want that. So we wanted some sort of a slow release kind of a chelating agent that would not kill the plant and also would not mobilize the lead too quickly. And so we landed on this chelating agent called EDDS. It is of fungal origin. And it is a chelating agent that can be applied to soil, and then its half-life is about 15 days. So after 15 days, it sort of biodegrades gradually. So we wanted to check out which one would be more effective. We hope that you could use something that is not as potent as EDTA. And we collected soils from 10 homes each in 2 different cities, which were known to be lead contaminated. And so the 2 cities were San Antonio, which was where I was at that time, and then Baltimore. The reason we chose Baltimore to compare with San Antonio was that San Antonio had soils which were highly alkaline. And so we wanted to contrast that with Baltimore, which we knew the soils would have lower pH. So because pH has a big effect on how mobile the lead is in the soil. So I'm not showing any data from our lab incubation study, but what we were able to do in the lab is that demonstrate that EDDS is able to mobilize lead, number one, but not as much as EDTA. And we also sort of looked at what concentrations of these chelating agents do we need to use when we go to the greenhouse. And so we landed on certain concentrations. And after the laboratory incubation study, we went to the greenhouse study, which was our phase 2, where we brought in the actual plant. Before that, we tested whether the plant is able to tolerate lead or not, how much lead is it taking up, all of that. And then in the greenhouse study, we use these columns. These are made out of PVC pipes, which we cut, and we prepared these columns ourselves. So you can see the column design. The column is about 15 inches tall and 6 inches in diameter. At the bottom, we have just clean sand, which doesn't hold on to any lead. But on top, we have the lead contaminated soil from different homes. And then we grow the vetiver in the lead contaminated soil. And so the design is that we grow vetiver for about 2 months. And after growing for 2 months, once the vetiver is stabilized and it's starting to grow, then we use the chelating agents at these concentrations, 0, 5, 10, and 15 millimolar per kilogram of soil. And then once we apply it, on the 60th day, then we wait another 10 days, and then we collect soil and we collect the plant. And of course, we also collect the leachate at the bottom to see whether any of the lead is leaching out, which we want to minimize as much as possible. So from the greenhouse study, we sort of found that the EDDS it was almost doing as well as EDTA when it came to the Baltimore soil, where the pH was low. In terms of mobilizing the lead and the vetiver removing the lead from the soil. In case of San Antonio soil, which had high pH, it was a little more difficult for the plant to take up lead. So that is something we realized, but it was still doing about 70 to 80% when compared to the Baltimore soil. So we thought that EDDS is something that we want to move forward with. So that's what came out of the greenhouse study, was that not only is vetiver grass tolerant and able to take up lead, but also that EDDS would be a good chelating agent to use. So now we sort of implemented a study, which was the phase 3, where we took the whole thing outside, exposed to the elements, but we were still not growing the plants on the ground, but we still wanted to control any kind of leaching or runoff, and then test how much lead is being removed. So these planter boxes were built. So we got them built with a particular design in mind. So they are inclined a little bit. So when there is rainfall, we could collect the runoff through these tubes here. And then there's also, you can see a little faucet over here where we were collecting the leachate from here, from watering as well as from the rainfall. And there's a tank at the bottom which collects the leachate. And so again, we had clean sand, about 12 centimeters of it. And then on top of that, we had the lead contaminated soil. And so here you can see a postdoc, Dibs's postdoc, actually, he is spreading the lead contaminated soil over here. So we grew vetiver grass on these panels, as well as we grew, just for comparison, a common lawn grass like fescue grass for 3 months. And after 3 months, we did our first EDDS application. And from the greenhouse study, we decided that the concentration we want to use was 10 millimoles per kilogram. And so that's the one we used. And then second application we did after 13 months. And there we did 30 millimoles of EDDS. And then, of course, we collected soil, plant, and leachate samples, and then we tested them. So here I'm showing a few pictures. You can see how well the vetiver grass is growing over here. Here's a student watering the plant. And then fescue, of course, you have to grow from seed. So here's a student spreading the fescue seeds on the planter box. And here is the EDDS application. So the application is done after 3 months, like I said. And here's some data that I'm showing, where you can see here I'm comparing the vetiver with the fescue. And so you can see that after 3 months, you can see that 3% of the lead is removed. And then before 15 days, and then after 15 days of EDDS, another 3.3% of the lead is removed. And then this is after 10 months. And then after EDDS application, another 3.2. So overall, about 10.5% of the lead was removed through this two times application of EDDS. Whereas when you look at the fescue grass, only all these steps considered, it was only 2.9% of the lead was removed. So now, looking at the condition of the vetiver grass and the fescue grass, however, you can see that vetiver grass is still doing very well. You don't see any toxic effects. The vetiver looks healthy. And then you can see the fescue grass is almost dying. It's brown. So definitely you see the toxic effect of lead on fescue grass. And you do see that it removed only 3%, whereas vetiver removed about 10%. So, and then the other thing we wanted to check was, does the grass prevent erosion from the platform? Because these planter boxes are pretty big. How much of the soil is eroding out? Because the roots are supposed to hold on to the soil, right? So we can see the control where there was no plant. This is the erosion. It's milligrams per kilogram. And then you can see for fescue grass and as well as for vetiver, actually both were doing pretty well. Vetiver slightly better in preventing erosion. And another set of data I want to show here is the composting of the vetiver grass. So what we did was we took the grass clippings and we wanted to see whether is this biomass, is it hazardous? Does it have to be disposed of as hazardous waste or not? So when we composted the vetiver grass as well as the fescue grass, and then we did T-clip on them, that is, we try to leach out the lead and see how much lead is leaching out, then you can see that in both cases, actually the lead leaching is minimal. So according to US EPA, about 5 milligrams is the limit that they look for. So if you are below the 5 milligrams per kilogram leaching, then that is acceptable by US EPA. So you can see that the leaching levels are pretty low, about less than 1 milligram per liter. So after our 3 steps, this is sort of the summary of the study. That is, number one, what we found was that vetiver grass was really effective, did not show any toxicity symptoms, and along with the chelating agent, it is quite effective in removing lead. Compared to fescue, there's no comparison. Vetiver accumulated 35 to 50 times more lead. And the decomposed clippings that we looked at, we saw that there was not much leaching from those. So when we did a T-clip, the leaching of the lead was significantly lower than the US EPA limit. And so conclusion is that we could possibly dispose of these clippings safely as non-hazardous waste. So now we thought that our technology is at a stage where we could do field implementation. So that's the study I'm going to describe next. So here we chose 3 houses in San Antonio and 3 houses in Jersey City to do this. And so you can see the first stage, what we do is that we do a grid sampling of the houses to see where the lead hotspots are. And for doing the grid sampling, we use a handheld XRF machine. So you can see a student looking at the lead. So this can be taken to the home and you can get the lead levels right away. And when we compare this to actual laboratory data, they are highly comparable. So this is a good instrument to use to get instant data. And so you can see the range of lead concentration in San Antonio soils versus the Jersey City soils. Some of the soils, they have really high levels of lead. So the US EPA limit is 400 milligrams per kilogram. So you can see how much, how the levels are pretty high over here. So, and then we also, in addition to 3 homes in each of the cities, we implemented this in a community garden in Jersey City. So you can see that the community garden, we also planted vetiver, and here we faced a particularly major issue, which was unforeseen, was that this particular building over here, this building had lead-based paint, and it was actively, there was construction going on, and it was actively leaching out lead and also sending lead dust to the community garden, which of course the users of the community garden did not know. And we only found out because we were conducting our study that the lead levels were going up and down and they were going haywire for some time. And so that was something that was a huge realization for us, that construction activities can really contaminate surrounding areas. So the design was that we used the vetiver treatment. Wherever I say treatment, that means we used the chelating agent EDDS. When we say vetiver control, there is no EDDS. And then we used a grass, Bermuda grass this time, without any EDDS. So here there are students planting the grass in the community garden. And here's some data that you can see. So overall, I'll just go very quickly on this one. So 3 years of implementation, the vetiver treatment plot, meaning there was a chelating agent here. And the important thing is this level after 3 years is below the 400 limit, which is what we are looking for. The vetiver control plot, it also sort of went down, but this is without the chelating agent and it is not below 500, but it does overall go down. And you can see these spikes here and there. It's because of this active input from the building next door. And then the Bermuda grass, you can see that it really doesn't, it did not sort of lower the lead levels at all. So here are some more pictures of students. So this is a house in San Antonio where the yard is being prepared first, and then the planting is being done. And then here you can see the fully grown vetiver. It's being it's, of course, we fence it off so people don't get into the hotspot area. And then these are the this is the data from the actual homes. So again, you can see overall that the lead, we were able to reduce the lead to below the 400, which is our goal. In the treatment, of course, the control, again, there's hardly any reduction. This is in Jersey City, and then this is in San Antonio. And then again, you can see that we do come below the below 300. But we started off with lower level of lead here. That is San Antonio. And then lead levels in the grass itself, you can see that, of course, you do expect that lead levels go up. This is Jersey City and this is San Antonio. and in the plant tissue. So that is expected. So overall, summary from this chemically catalyzed phytoremediation is that we are able to design and implement a particular method that we feel is easy for the homeowners to implement themselves. So we sort of have a step-by-step program that homeowners could implement. We used a chelating agent, which is biodegradable, and that chelating agent is able to mobilize the lead so the plant can take it up. So that was sort of the major achievement. So this is something that when I go to either conferences that HUD actually organizes with some of the people who are either non-governmental organizations or practitioners or lead inspectors that work with the people who live in these lead-contaminated homes, and we sort of give them this steps that they could follow to implement this in their houses. So so if they call us, we go, we could do a grid survey of the property and then collect the samples. We do a lab test to see what level of chelating agent is needed. So that is called a CAO test that we do. And then we could recommend the amount of chelating agent they need to use and how frequently do they need to apply the chelating agent. And then they could plant the vetiver grass, and then after 3 months they could start spraying the chelating agent, and then of course collect soil, send it to us for testing from time to time. Vetiver being a perennial grass in places like San Antonio in the south, they'll keep growing, they'll be dormant in the winter, and then they'll come right back in the summer. In places like Jersey City or in New Jersey, if there's a lot of snowfall and the ground freezes, then some of the vetiver might die. And then come spring, you might have to replace some of these plants. So I don't know how much time I have, Tibbs, to go over very briefly the
Yeah,
next
go.
method.
You can go over briefly. We still have about 20 minutes. So I'd like to have at least 5-10 minutes for Q&A. But
Okay. So this is the next sort of method that we are developing in Deuce's lab and my lab, is that we are trying to look at chemical immobilization. So this is the other approach. The first one was phytoextraction, right? So we were using plants to remove the lead. In this case, chemical immobilization is that you leave the lead in place, but you make it bound very tightly, and so it's not available for uptake by the plants, and it is also not bioavailable. So reduce bioavailability, reduce plant availability using chemical immobilization. So this is the second sort of method that we are developing, and we are in the beginning stages of this. So the idea behind this is that this is sustainable because we are using amendments such as biochar. So biochar in itself, it is widely used by farmers for soil conditioning, and it is also known to bind metals. So we could perhaps use this to bind the metals so that bioavailability, plant availability is reduced. So the main thing that we don't know is, we do know biochar binds metals, but what is the long-term stability of the bound metal in the environment? That is the main question that needs to be answered. And so this will again, of course, depend upon the soil properties, the environment and all of that, the climate. And so we want to go deeper into this and see how effective this is in the long term. So what is biochar? You must have all heard about biochar. It is just made out of heating the biomass. It could be agricultural biomass. And it's heated without oxygen, and you get the char, as you can see in this picture. And so it's widely used anyway to improve soil pH. It adds nutrients. Also, it acts as a carbon sink. It increases water holding capacity of the soil. It reduces soil erosion, nutrient leaching, and more people are proposing to use this for remediation of contaminants, for waste management, and of course, because of the carbon sequestration, for climate change mitigation. So our experimental design is that here now we have collected soils from 30 different houses in 3 different cities. So we have Baltimore, Detroit, and San Antonio. So Baltimore and San Antonio we chose because we've been working with these soils. We know them inside out, and we have a good idea of how they behave. And then Detroit, because I'm in Michigan, and Detroit is a major city where there's a lot of lead contaminated houses. So we have introduced this one. And so we collected these 30 different soils. And then DIBS's lab has been working on initial soil characterization, and then amending with biochar and looking at how the immobilization is working. So our idea is that we want to see how the lead is binding with use of biochar. We want to see whether bioaccessibility is getting reduced because of this. Bioaccessibility is basically bioavailability. You do a beaker test to see whether bioavailability is getting reduced. And then another way we are checking for bioavailability in my lab in Michigan is looking at human intestinal epithelial cells, because we are sort of simulating the ingestion pathway where children would put their hand, play in the soil, and then put their hand in the mouth, and how much would they ingest, and then what happens to the lead in that soil. How much of that lead is bioavailable? So we are using intestinal cells to check that. And then once we get an optimal sort of rate of biochar and an optimal biochar, then we will do greenhouse studies to see how effective the biochar is. So that's the whole experimental design. Now, the initial data, this is just a little bit of data that I'm showing here, where you can see this is sort of a little bit of a complicated figure, but this is something, it's called sequential extraction of the soil. So what we do in sequential extraction is you take 1 gram of soil and you extract it with different liquids. And then you see where is the lead bound, okay? And depending on that, how stable is that binding, okay? So that's what it tells you. So here you have the F1, which is the soluble and exchangeable. This is the one which is bioavailable, as well as it is plant available. So this is the one which plant will be easily able to uptake, and this is the one which we've been trying to solubilize using the chelating agent. So you can see that a lot of it is in the F1 fraction to begin with. The second one is the F2 fraction, which is carbonate bound. The third one is the F3, which is oxide bound. F4 is organic bound, and F5 is the residual silicate bound. I'm sorry. So you can see little bit is silicate bound. Most of it is soluble and exchangeable. Some of it is oxide bound. Some of it is organic bound. And then after applying biochar, within 7 days, you can see that there's appreciable decrease in the soluble exchangeable part, and there is increase in the residual part. So this is what sort of the take home that I wanted to show you. I'm not delving into detail. So we are doing a lot of permutation and combinations of biochar application to see what is the effect. And then the next thing is we are looking at actual bioavailability and bioaccessibility to see how much of the bioaccessibility, how much of the lead is becoming bioaccessible. And then we'll be doing these incubation studies for various periods of time to see how long does it take for the solubility, accessibility to be reduced. So with that, I'd like to acknowledge, first of all, the US Department of Housing and Urban Development. So they have a lead technical studies program, which has been funding all these studies right from 2004. Continuously, so we are thankful to them. All the homeowners that allowed us to come in and do this work in their homes in San Antonio and Baltimore, we are very thankful, especially through the COVID period. We also continued this study. And then we liaised with a nonprofit called Green and Healthy Home Initiative, and they are the ones that are our liaison with the homeowners. And then Willie Villarreal, he's from the Neighboring and Housing Services Department in San Antonio, who helped us, and Anna Pinter from City of Detroit. She is also sort of a liaison for collecting these soils. And then also all the people that have been involved in this study right from the beginning, I'd like to thank them. And with that, I'd be happy to take any questions.
All right, that was an excellent presentation, and as you can see that the board is